Hi, everyone. Welcome to our poster. We're talking about the interpretability of network measures and host virus protein interactions. So the project goal is this. Can the analysis of virus-host protein interactions identify critical host factors? So say you have a virus that's infecting a host. So we can look at this interaction by looking at the protein-protein interaction networks, which each protein is a node and an edge between them denotes an interaction between those two proteins. So we have the virus proteins as triangles and the host proteins as circles. So during this network analysis, we want to know if we block protein 1 from the host, is it going to prevent viral replication? And we're going to look at this in case 1, where we block off host protein 1, and case 2, where we block off host protein 2. So where are we getting this data? Well, we're getting this data from viruses.string, which is a great database. It has a lot of protein-protein interaction networks between viruses and their hosts. And what we did is we downloaded all that data and we only kept edges that were experimentally verified. There's a lot of other edges that are from text mine data or from inferred edges, but we're only looking at the experimentally verified ones for now. And then after we have those networks, what we're going to see is we're going to perform all of these network measures and look at all these node level analyses to see, okay, given these particular network measures, is there a relationship between high or low value and a physical property about that protein? So we looked at all of these different network measures, which are node level measures. And you can see some of these examples here with degree centrality and the yellow one between the centrality, eigenvector centrality, where the yellow nodes are high values and the gray ones are the low values. We also looked at the whole network measures as well. So you look at the average node connectivity or the average node centrality for the whole network, or looking at the Shannon-based non-randomness or a Kolmogorov-based approximation of the complexity of the network as well. So what did we get? So in this data set, we got 55 networks, and that had a significant amount of edges, more than one edge. The largest was HIV, and the other ones, the other largest ones are, of course, human virus pathogen interactions. So we looked at some of these measures. This example is the eigenvector centrality node value, which is the third cartoon here on the previous slide. And you can see that nodes are going to have different values of this measure. So you can see that there's one bright yellow triangle, and then the rest of these are circles. And then there's another yellow triangle as well. So what we want to know now is we want to be able to compare networks to each other, meaning, are there any networks that have a distribution of measures that are different or a distribution of measures that are the same? During our first pass, actually, we didn't really find anything that interesting. In order to compare these different networks, we looked at the rank order of these different node values. And when you look at the rank order, you get one curve is going to correspond to one network. Well, the good news is, well, I guess it's not really good news, but one thing is that all of the different shapes of these distributions were fairly much the same, about approximately the same shape, but that's actually common to these biological networks, so we didn't find anything here. So we won't be using this as our approach moving forward. But what's more important for our approach is this idea of making sure that our networks are the ones that we actually want to be looking at, because we're missing a lot of data from viruses and hosts interacting with each other, but also our networks that we currently have might have nodes and edges that we don't actually care about during the infection process. And because these network measures rely so heavily on the network topology, it's really important that we get them right. So what we're going to do is we're going to use a prize collecting Steiner forest method in order to get the correct subnetworks, at least the subnetworks that are the same throughout each network, because some networks might have a lot more nodes because they're just simply more well studied. So how do we do this? Well, we're going to use proteomics data. And what we're going to say is during this infection process, does the expression of a particular protein go up, or does it go down? And if it changes, goes up or goes down, we give it a high score, so like this yellow here. And if it doesn't change at all, then we give it a low score, like this gray node over here. So once we have those nodes scored, the price collecting Steiner forest algorithm is going to infer a set network, which is basically the smallest possible network that will have all of these high level prizes on it. So it's going to have these subnetworks that are containing proteins that are the ones that are changing during this infection process. And so once we have that, we're going to repeat our analyses by measuring all of the different network measures on all of our different networks. And then what we're going to do is we're also going to include physical data about physical properties of the proteins, compare them, and see what happens from there. And our hope is that if we find trends and we find correlations between these network measures and a physical property about a protein, then we might be able to say something about how we can change the infection process, or we can even possibly reprogram the interaction. Thank you so much. Hope to see you there.